I'll tell you today about the part of our research in my group which involves use of tissue engineering methodologies to produce the systems that we could use for pharmacological testing potentially, as well as in the future we hope for therapeutics in regenerative medicine mainly. So talking about skeletal muscle, just kind of a little basic reminder. This is a muscle that comprises 40% of our body. And in general, if you look at any part of the muscle, you can see that it consists of very long cells. These cells we call muscle fibers. So when I refer to muscle fibers or myofibers, I'll be referring to long elongated cells that have many nuclei. They are surrounded by extracellular matrix and other fibers. And what I will show you today is basically engineering, tissue engineering of one of the bundles from this bundle, as shown here, is a muscle bundle. It's a group, actually, about 200-300 microns or more in diameter of muscle cells. And this is more in medical terms called muscle fascicle. But what we'll be doing and what I'll be showing you is engineering on one of these fascicles and then functional assessment and some drug tests. Okay, so alright, so why would one want to go with the human muscle and what is the whole problem? So the issue with the whole field, actually from about the last 15-20 years, has been that there has not been a functional contracting muscle. So as we all know, contraction is one of the most important properties of skeletal muscle. People have been able to make a rodent engineered muscles that would contract. But human cells, although they would be taken from the muscle biopsy, for example, from patients or from donors, would not be able to make into the working muscle. So why would one anyways want to make a working human muscle? So there are a number of reasons that I kind of singled out for. So first, there is a huge genetic diversity between humans, as we all know, as we can see among each other. And then that diversity is much less in the mice that are used in laboratories. So basically, the animals used in laboratory practice are much less diverse. And so this is one of the important things to work with human muscles. Severity of human disease, when you generate muscle models of disease in animals, are in general less severe than they are in humans. So again, we don't have a good disease models for using the animal cells. And then human engineered muscle actually would offer the ability to rapidly go through a number of conditions, because we will be able to make a number of small muscles, which is the goal, and then be able to test many drugs at the same time. Something that saves the time, saves the costs, and is actually a powerful way to pre-screen and test drugs in preclinical practice. And then we can actually personalize these tests, meaning that we could take the biopsies from a particular patient, generate these muscle tissues, and then be able to look at the personalized responses of these patients to particular drugs. Okay. All right, so how we do that? So we actually go and isolate the small, like a green pea-sized piece of muscle from humans, from a biopsy, and then we dissociate it using different enzymes into single cells. And then we can use these cells and expand over multiple times, 50,000-fold, 100,000-fold, and so on. And after we get millions and millions of these cells, we can use them and coax them back into these big muscle fibers. So I've shown the muscle fibers and cells how they look, and basically what we start with is these single cells. Here shown, each blue dot shows the nucleus of a cell. And so these are the muscle cells that we get and we use them to engineer muscle. And so if we do this just on a dish, on a plain dish, culture dish, we end up in generating muscle fibers or things that are these elongated fibers by switching these to a differentiated soup of ingredients that will differentiate these cells to become muscle cells. Now that's possible to do on a dish. The issue is, again, as I mentioned, that this will not contract. Right? So with human cells. Okay, so recently and over the last probably 20-25 years, the tissue engineering methodologies have been developed to basically make a three-dimensional culture. So instead of putting the cells on a planar dish, putting them together within the volume, right? And trying to make these cells work together within the volume. And what we do basically, we dissociate these cells and then prepare these molds which will harbor the matrix in which these cells are, that we call hydrogel, basically like a jello-like matrix. And then we will dissociate these cells that I've shown from the muscle biopsy, expand them, get a lot of cells, mix them with a hydrogel, and the hydrogel is a three-dimensional matrix dimension. Is very much similar to, in essence, to blood clot. So this is the same material that you find in a blood clot. It's called fibrin, right? And then you can use these cells, mix them together, put in these kind of molds, and over time you will see that these cells come together and form what's called engineered muscle bundles. So these two bundles here are basically three-dimensional tubes, or like muscle fascicles, that consist of fibers inside. And I'll show you how they look inside in the next slide. Yeah, important thing to mention is, so we get a very small biopsy, so the green pea type of biopsy, 50 milligrams. We dissociate cells, then we expand it. We're able to get about 1,000 of these little bundles from just a little piece of muscle biopsy. So that allows us basically 1,000 independent experiments that we could do with a particular drug. Right, so when we look at this, and this is one of these shown in the culture. Okay, so what we see is basically that they will spontaneously contract. So these are the twitches of a muscle within this configuration. It's spontaneously twitch, and these are just different examples. And so this is actually the first time I observed twitching of this muscle. So we actually looked then what's happening inside of this, and we tested them further functionally, and I'll show you how. So just so you see how this looks inside. So this is the muscle bundle shown. There are different types of cells here, but predominantly the green colored cells are muscle cells. So these are the muscle fibers that they describe, and surrounding them is a sheet of fibroblastic cells, which are basically connective tissue cells. And the fascicles that they mentioned initially in the original muscle, in the native muscle, also are surrounded basically by the thin layer of fibroblasts. Now inside, if we kind of zoom in that, within that muscle fibers, we can see cells that are aligned. They have multiple nuclei, and this is characteristic of muscle fibers. They are cross-striated, and cross-striation again is a characteristic of muscle fibers. And these are just basically different markers that we use to show. These are the cells just shown in a cross-sections, so they're basically coming out of the plane, and they're in parallel directions. And this is again additional staining, just confirming that cells within these myofibers, within these bundles, are differentiated. So we get something that's a differentiated muscle. And if you look what happens with the size of these fibers with time in culture, this is now, we're culturing these for 4 weeks, you can see how they're increasing, so the diameter of these fibers increases. In general, the diameter in an adult person versus just an embryonic or muscle, the diameter of myofibers increases significantly. So when we look at this in culture, we would like to see this differentiation process to happen and to see the increase in fibers, and this is kind of a signature of that. And then we can look at different proteins, and I don't want to go into this, but in general, all the muscle proteins that we study, muscle-specific proteins, will also be increasing during time in culture. Okay, now the most important question is, if we electrically or chemically stimulate these bundles, can we see the contractions? This is that missing thing that I mentioned was basically hampering the field for the last 15 years. And so if you stimulate these, and so what I'm having here is a frame made kind of a semi-elastic, kind of a pliable frame, to which these fibers are attached. And this is stimulated electrically at once a second, 1 hertz. And you can see the contraction of these fibers. So basically the fiber of these bundles, so the myobundles, human bundles, can contract. This is the first time that has been done. Okay. So what it allows us to be able to do all the physiological tests in a dish. So all the tests that we normally only been able with human muscle to be done on a patient, in a live patient, now can be actually taken in dish and performed. A different test can be performed. And one of the tests, just to kind of prove that this is indeed a physiological system, is to apply either a single stimulus, as shown here, or apply multiple stimulus at different frequencies, 20 hertz or 40 hertz. And what you can see is that this single stimulus twitch, when you apply multiple pulses, fuses together into more sustained contraction that's called tetanus. And this is a classical way how our muscles work, through these tetanic contractions. Right? And so this is the same thing just shown in multiple experiments. Right? And also there is another property of the muscle that we know, that if we extend the muscle, if we stretch it, in general, up to a certain length, it will actually increase the force of contraction. And again, if we measure this in this system and keep increasing the length of a muscle, we'll see the increase in the force. And that actually is, again, a well-known physiological property of the muscle. So, what happens during the time in culture? Well, you know, this is kind of, again, the single twitch, and then these tetanic contractions, and the amplitude of it shown over the 4 weeks of culture. As you can see that basically with time, in general, we have an increasing trend. the force that these muscles can generate. So that's been one of the things we want to do with this. Another thing was basically everything I showed you was from one biopsy, from one single person, right? But of course, the goal is to make a platform to be able to get this from any person and then be able to generate these types of muscles. Well, this is so what we've done. We actually isolated these muscle biopsies from 10 independent individuals. And actually, for now, we repeated this in over 30, almost 40 individuals, as a matter of fact. So basically, D1, D2, D3, these 9 donors, different types of donors, and just different individuals from which we would, again, get the cells, get a little biopsy, expand the cells, put them together in these muscle bundles, and then study the force of contraction as a sign of functionality. And so these are the, basically, amplitudes of these forces, and you can see the variability, about twice variability in the amplitudes, and this is actually, a variability that we would expect to be in human, different, cells from different donors, right? So, okay, so this is one thing. So the system appears to be robust because it actually applies to many, many different donors. Now, one thing, if you want to study drugs within this system, is to be able to to monitor how the system works, but every day without really destroying it. So one way to monitor the system would be to take this bundle, test function, and then histologically section it and see what happens and so on and so forth. But if you want to be able to study something online, basically in real time, you need to bring an indicator or function of some kind that you can assess in a non-destructive basis. And so for us, that's basically this genetic indicator of intracellular calcium concentration. So every muscle twitch or contraction is associated with a calcium spike in the muscle, and this is how the contraction happens. So if you provide a special protein that contains a GFP molecule and the calcium binding molecule together inside these tissues, you'll be able to actually monitor when they have a calcium spike and when they contract, just by looking at the fluorescence flashes. So technically you can go inside that system online and just check the fluorescence. And I'll show you examples of this. So this is basically during this pontaneous twitching of these bundles. This is the bundle is positioned this way, and what you can see here and there are just twitches of a single cell is happening randomly, which is kind of classically like a muscle tone in general is generated through these types of contractions. Then, if you stimulate this at 1 Hz, for example, as shown here, you can see how a lot of these muscle bundles will now trigger calcium and contraction, synchronously. And if you further increase and apply 10 Hz, now you can see a much higher calcium level, and this is what we're doing the tetanic contractions. So we tested this, and this is basically the same data shown here during the 1 Hz or tetanic application of calcium. You can generate and look at the calcium transients. And with time of culture again, the calcium transient is a sign of maturation of this muscle increases. So what I'm not showing the data, but we basically then compared on one-to-one basis how this calcium amplitude and the force amplitude correlate. And we found a really good correlation between the bundles in which we measured the function by measuring the force they can generate and the calcium amplitude. So basically this could be used, looking at the calcium could be used as a surrogate of the contractile function of the muscle. So then we just went to kind of validate this muscle with a couple of drugs, right? And so this is one example where, as we all know, if you add the calcium, drink caffeine and too much coffee, you start to become jittery. So what we did, we basically applied a bolus of caffeine in these bundles, and this is one of them. And then we look what happens with time, and as you can see, the calcium levels and little twitches within this bundle will start slowly to build up as we put the caffeine in. And you can see how your calcium levels and tremor kind of starts like increasing. And so this is an example of that, and this is just the same thing showing the increase in calcium, as well as force of contraction with application of caffeine, and just kind of like multiple donors, because all of the experiments we do, we usually do at least 3 or more donors to just confirm that this is not an individual response, but rather more of a group response. Okay, so one other kind of a way to look and evaluate the functionality of these muscles is to look, a classical response to acetylcholine. Acetylcholine is just a neurotransmitter that's released by our nerves, and then it engages or makes our muscles contract. So normally we don't have electrical stimulation, of course, in our body, as I've shown, but we have a neuronal release of the transmitter, and this is acetylcholine. So we want to do this bundles without neuronal stimulation, are they still responsive to neurotransmitters? And one of the things to look at, and one of the important parts of this whole neuromuscular system is the acetylcholine receptors shown here in green. So our muscle fibers express these receptors, and then we apply a bolus of acetylcholine, like almost mimicking the neurotransmitter release. You'll see that at some point it will saturate the calcium, so basically will induce the calcium response and contraction in this muscle. So this muscle responds both in terms of electrical responsiveness as well as they have a natural chemical responsiveness to acetylcholine. So I mentioned in the beginning that the whole idea of this system is to be able to try to test drugs and look at the drug toxicity or drug efficacy in this system and be able to do it at as much of a high throughput fashion as you can. So some of the things that we want to look at are classically known drugs to induce damage or toxicity to muscle. And some of them are even withdrawn from market. And examples of the drugs are classical statin drugs. And statins are cholesterol-lowering drugs, widely used on the market, and are known actually to be myotoxic depending on the concentration. However, by mouse studies in mice, some of this toxicity could not be predicted, and in particular for some statins, the doses that were not toxic in mice turned out to be toxic in humans. An example of one of these drugs is actually cerebrastatin, which was withdrawn from market after the basically fatal rhabdomyolysis, the muscle disintegration has been seen in some of the people that use it. So lovastatin is another statin, but which is still in use, in wide use in among patients. So what we basically looked at what's been a dose response, what's the generated force of the muscle, how it changes if you increase the dose of a statin. And if you compare these two, cerebrastatin, look just at these doses. This is nanomolar, and this is micromolar. So these are basically 1,000 times larger doses here than here. With the increase in dose, in both cases, you have a decrease in force due to the myotoxicity. But cerebrastatin, the muscle that we see here, is sensitive, much more sensitive to cerebrastatin, since smaller doses are needed to introduce the toxicity. So what this means is basically that we are really catching the higher toxicity of this drug versus this, which is at the doses that are clinically relevant. And so this is something that has not been noticed in mice. So technically, this serves or could serve as a predictive test bed for looking at the toxicity of different drugs to muscle. And the interesting thing is that if we look at this and stain for the fat accumulation, in statins, especially after the toxicity is induced, it's known that a fat is produced. And so when we look, we actually see that at increasing doses of these statins, you start to see lipid accumulation within the muscle. So we see more clinical signs, histological signs of how these drugs act on muscle. And the second example I want to show is basically an example to responsiveness to Clenbuterol. And Clenbuterol is a drug that's not approved in the United States. It's used by bodybuilders to increase the muscle mass, and it's been shown that actually it can have adverse effects. And because of that, it's not FDA approved. It's approved for some applications in Europe actually. So if you start increasing dose of Clenbuterol and adding to the muscle, you can see both acutely, this is just basically for 30 minutes or over many days, which is shown here like a week or 2 weeks. You can see the increase, as the dose is increased, you can see the increase in the force generation. But however, for larger doses, you can start seeing myotoxicity. So at the lower doses, there is still some beneficial effects. At the higher doses, there is muscle toxicity. And this drug is actually kind of illegally said that they're called Clenbuterol tablets that people use for the bodybuilding. But obviously it does have damaging effects to muscle at particular doses, at higher doses. And again, this increase in force shown here is also, again, we confirmed it for 3 different patients. And if you look why this increase in force, why these muscles are becoming stronger, we see that basically by just looking histologically and doing some quantification, we see that there is some muscle hypertrophy. So this Clenbuterol is used for muscle hypertrophy, but we really see, we do see some, but as has been already described, it's not really that much useful for muscle hypertrophy. People realize more for the leaning of the muscle, in many ways become toxic after the at the higher doses. Alright, another examples I wanted to show is just basically exercise, use of exercise in the muscle. And of course, you know, these muscles now being functional and being able to contract, we can use it to look at the different exercise regimes. And in particular, the way how we would mimic exercise is just by electrical stimulation. So I'll show you just, if you apply electrical stimulation to these carbon electrodes, you can see basically how with the application of stimulation, these muscle bundles are twitching, and you can see that you've been able to see this motion. So we can use this and apply different regimes of stimulation, and then check like different exercise regimes basically, check what happens with muscle. And one thing that's obvious here is this is why you need to exercise. If you do exercise, this is a sedentary, kind of non-stimulated muscle or non-exercise, and this is what happens in different regimes of exercise. You can see there is at least 3-4 times increase in muscle contraction of the muscle. And if you look inside the bundles, this is basically the muscle fibers within this non-exercise control fibers, and these are the muscle fibers when you exercise. And obviously, there is a large increase in size, and there is a, you know, just a better structure of this muscle. And if you section the cross-section of the muscle, so you cross-section to the bundle, now these muscle fibers are going out of the plane. This is the non-exercise, and this is the exercise muscle. And as what you would expect, during the exercise, there is basically a retroperitoneal muscle, which is why people, among other things, why people exercise. And you know, we can quantify how each fiber is increased, or how the whole muscle bundle is increased, and there is a both increase in total mass of muscle as well as individual fiber sizes. Okay, so you could use this then to look at the different types of drugs for increasing the muscle mass, or actually preventing potentially muscle wasting, which is something that's common to many diseases. So I'll finish just with the example of use of this system to model a particular disease in the muscle. And so we're lucky, or here at Duke, to have a large cohort of patients with a rare disease called Pompe disease, and that's a disease which there is an enzyme called alpha-acetylglucosidase that's missing from the muscle and the whole body. And when the enzyme is completely missing, that's called infantile onset disease, and really there is a big heart disease that forms, and unfortunately babies die within a year or two. And then there is a so-called late onset disease where this enzyme is missing but partially, and the disease manifests itself about 30 years of age or 40, where the muscle weakness starts to happen, and then respiratory failure. Patients go to wheelchair, and then finally there's respiratory failure and early death. But recently there has been an enzyme recombinantly made, basically by Genzyme, by a company, and it's been added to infants and has actually been able to save their lives. But the drug itself is quite non-efficient. It's extremely expensive. It's about $300,000 lifelong therapy a year. It's a bi-weekly shots of this enzyme for intravenous shots for the kids and so on and so forth. So there's a lot of issues with it, And so I just want to show basically that you can use this system to model this disease. And so this is just an example of how the muscle normally looks. And if you actually look at the muscle when there is a disease, there are these purple granules in the muscle are actually glycogen accumulation. So because of the metabolic problems with this disease, there's accumulation of glycogen and finally muscle death. Okay. So what we've done, we basically, in collaboration with Dwight Koval, we've been able to get biopsies from these patients. Now over 15 patients, and we would do actually the same type of approach: dissociate the cells, put them in a culture dish, and then put them in these three-dimensional muscle bundles. And this is just an example of one of these after a 3-week culture, where if you look at the healthy muscle bundle, the one that's made out of the normal donor cells, there is really no red accumulation of glycogen. But if you get the muscle, the cells from the patients with late onset Pompe disease, about 40 years of age or something, you start to see accumulation of this. So there are structural kind of things that are known for this disease that happen also in this injured muscle. And this is another example of protein called LAMP2, which is a lysosomal protein, and the feature of this is an accumulation of this protein in the muscle cells. And indeed, after 4 weeks of culture in the healthy muscle models we don't see this, but in the disease model we start seeing accumulation of 1, 2. So which is talking about lysosomal accumulation. So functionally, again, normally we see an increase, as I've shown before, with the time of culture of this muscle. But most of these muscles, now we have more data, actually with time of culture the function decreases. So there is a there is a deficit on the functional side, and it's a muscle weakness basically that's known to exist in these patients. So what we are now working on is basically doing a drug testing for a lot of different drugs, and in collaboration with DTRI, also here at Duke, we are looking at parallel trials in clinics with these patients where they are tested for new drugs, and the muscle biopsy that we take from individual patients, and we test the same drug and more drugs in this system. So we're able to basically, in a personalized fashion, to say if there are non-responders to a drug that's tested in clinics, does that happen in dish when we take those cells from that same patient? And then can we look at some other drugs that would be helpful for a patient? So this is kind of a take on really looking at this personalized medicine or precision medicine, as been recently coined or termed, and looking to see if we could help individual patients. And I'll just stop here and acknowledge people that really helped and made this happen. These are the students from my group and postdocs, and this has been all teamwork with multiple people from Duke, and in particular George Jersey here, also Bill Krause from cardiology, and then Dwight Coble from pediatrics, and other people who were helpful very much in providing the muscle biopsies.
So we have some time for questions. I wonder if our responders have any initial questions they might start the conversation with.
Thanks, that was really interesting, thank you. I can see quite a lot of applications, particularly for earlier stage, to try and avoid the need for lots of clinical trials early on, and to perhaps encourage innovation and trying out new therapies and healthy dinners. What I was wondering was, you talked here about using biopsies from patients, either in a disease state or healthy patients. Have you thought about using, cell engineering, where you're forcing genotypes and phenotypes, so that you can perhaps use these as more across the board models, where you don't have to rely on patient biopsies?
Yeah. So this basically is a methodology that would be made based on the induced pluripotent stem cells or pluripotent stem cell work, where you can just have a blood sample, or you have a general platform, or you get a fibroblast from somebody, and then go and reprogram these cells into the cells that can become any cell types, including muscle. So there were like 2 slides that I skipped there, which is exactly about that work. So yeah, so we are working on basically, taking this into ability to not depend on a muscle biopsy, but rather have, access to some easier accessible tissues, and even the cells, that types of stem cells that I'm mentioning, are actually also could be made from even samples of urine. So very non-invasively, basically, and then used to make into muscle cells. So the methodology to make these cells in muscle cells and especially functional muscle cells, is tricky. So it has not been yet done. So part of the work is basically trying to really make this into functional muscles, starting from these kind of sources, such as very neutral cell types, and ending up in a muscle cake. And we've been doing that, and we have some first progress, and are looking basically into making that to be comparable to what we get from muscle biopsies. We're working on that.
Beautiful, beautiful work. I'm a big fan. But following up to her questions, really, I have 2 questions. From one biopsy, how many construct can you make? And the second one is there data that proves that for measuring these drug responses, like the lovastatin and the seristatin, you need to have this beautiful 3D construct that contracts so that muscles in a 2D dish with the conventional endpoints do not pick up that dose dependence?
Yeah. So 2 great questions. So I kind of mentioned one slide that from a 50 milligram biopsy, which is really a very small biopsy, we can get about 1,000 of these bundles. So basically, this is 1,000 per one little biopsy. We could re-biopsy the person if needed, but also, we get also larger surgical samples based on different types of surgeries. For example, scoliosis surgery in pediatric patients is often a source that we also use to generate this. Now, there we get about 20 to 40 times more muscle than 50 milligrams. So the amount of these muscle bundles that we get is almost unlimited. So there is a potential to test many drugs, in the same, basically from sample from the same person. For the second question, yes, so that's a good question. So the tests that are dealing with function are the tests that are uniquely, suited for this system. We've also been looking a little bit, and we have some indications that the differentiation, the maturation of cells that's achieved, especially over long-term culture, right, that we can do in this type of system, is higher and is more advanced than what you would get in two-dimensional systems. Also, two-dimensional systems will often basically detach after just a week, 2-week culture. So this allows you actually longer-term maturation in a more natural environment. So all functional tests can only be, like contraction tests, physiological tests, can be only, tests of muscle weakness, for example, can only be performed in this system because the other system doesn't even contract. But also the maturity and with that physiological relevance is increased when we have a longer-term culture that's only again possible in this kind of three-dimensional system. So there's multiple advantages. There are also some of the things where you would be able to see probably also in just the two-dimensional systems, especially biochemical things. So this allows you also physiological tests, which is the most important advantage.
Really fascinating work. Some questions I had were, how does it compare with native tissue? So, is the contractile force similar, and is the response to drugs?
So it's a good question. So we look at in general compared to contractile tissue, we look in what's something that's called specific force, which is how much force is produced per cross-sectional area of muscle. And so in our best cases, when the muscle is exercised, we reach to about 30 kilopascals, millinewtons per square millimeter of tissue. What's normal in human tissues is more about 120 to 150. So we're talking about a 4 to 5 times lower specific forces, compared to the normal, to the regular human muscle. Now, most of the physiological responses that we looked into different drugs and like to the length increase or frequency increase are similar quality, similar with the human original, the natural muscle.
And also, have you done any work looking at contractile diseases like atrial fibrillation?
Well, for atrial fibrillation, we would need to go into cardiac constructs. Now we do have a parallel, it's parallel research in heart muscle and human heart muscle engineered tissues. So in those, we can study some of the cardiac diseases. Yeah.
When you're talking about the time to maturity for the 3D cultures, what is that timing? Because if we think about application, for instance, I would think that they'd want an answer about the drug response sooner rather than later.
So after 2 weeks of culture, we already get a relatively stable system. So all our drug tests have been done between 2 and now up to maybe 5-6 weeks of culture. So basically, it takes about 2 weeks to get a testable system.
And well, from a pharma standpoint, I'm sure you're thinking about it. What's the throughput you can achieve with this system? Can you do 12 at the same time? Can you do 58, 96 potentially? What would it take to go to 96?
So we don't have a system in place to measure multiple force responses simultaneously. We're working on kind of surrogates of that. Now, we haven't yet done, but it's possible to miniaturize the system into probably, I would say, maybe 48 well format. The issue is being able to miniaturize but keep functionality. So we haven't tried it. These are relatively small, so these are basically 7 millimeter or 8 millimeter in length, and so they will fit, as a matter of fact, in maybe on 24 well plate, in hollow plate for sure. So we could go probably look at the simultaneous imaging of this calcium proposition. That could be done simultaneously on multiple plates, I believe. Functional tests, you will need to make a different systems for testing than what we use. We use a classical force transducer to measure exact force. The systems that can be used as a high throughput are those that would take basically would rely on the deformation. So we could have some type of elastic posts where this would be deformed, and by basically calculating that, look at how much force they generate. So this would be optical measurements rather than true physical contact measurements. So they need to be optical after all, right, for the high throughput. Yes.
So the main focus of this talk was all around modelling, testing, and that kind of thing. Have you done any work, or are you thinking about doing work to actually look at tissue engineering for transplants and that kind of thing?
So we've done a lot of rat muscle work that kind of preceded this work, which helped us to know what we need to do to get to the human functionality. So we're looking implanting these, and we've done implantations in the small models. There is still a huge limitation for the large muscles in humans, which is there's no way to build a vasculature inside to be able to really grow a large piece of muscle. For applications such as facial muscles or lip muscles or some of the smaller muscles, this probably would do at the current stage. But for any kind of large muscle repair, there's a very important obstacles in terms of vascularization that you need to kind of resolve, and the whole field is working towards this. So we've done some preliminary implantations in small animal models where these small muscles can do some benefit. But for the large human-sized muscles, there's still a lot of obstacles to be overcome.
Do we have other questions from anyone else in the audience?
I was wondering if you might be able to propose a hypothesis of why the cells would have a lower contractile force compared to normal human cells.
Oh yeah, so there's a issue likely in the size of a muscle fiber, the density of the fibers within the muscle. Muscle fibers in cross sections are really dense, so almost maybe not 100%, very dense. So these are more sparser. So per unit area, there will be more force generating units. And then the contractile proteins and likely calcium signal on the inside are still immature compared to what's really achieved in a adult muscle. So these are the kind of main reasons why.
So would that have any effect in the testing, the density or the calcium maturity?
Well, in general, it gives you a smaller quantity, but since we have a really sensitive measurement device, we can measure the changes. Now, will that give a difference in some of the responses? We're testing that. So together with George and some other people, we are really going into a larger number of drugs to understand whether we would see a qualitative difference from what's known to existing clinics. So far we haven't really. So basically, qualitatively we believe we are getting the same responses, which means that the general physiology is there. Not maybe the quantity level, but definitely the qualitative level.
I wonder if you could comment on whether this could be used to produce food.
That's a good question. There are people that are really think about this. Not from human cells, but from different types of muscle cells, and where so far that would be probably, if you think about your steak, made of these cells, it would probably be at this point about $30,000, which and would be pale white. No blood, no fat so far. So so it wouldn't be a tasty steak, but there are actually there were a couple of exhibitions where they did form a burger. Not really steak, I guess. You can put anything in burger and it can look good. Yes. So there've been actually proofs of concept, very costly ones, that you can generate a small paste of tissue engineered meat.
Final question.
Just more of an industry-related question. So how do you envisage this kind of technology being commercialized properly? So I saw on the last slide you said Pfizer was in some way involved, but what do you think the steps are?
So so there are. this system by being functional, basically being able to do physiological tests. There are ways to. Obvious ways to study particular drugs in this system for their efficacy and for the mode of action. And some of the, for example, we're finding now that some of the ways how this engineered muscle feels glucose, which could be related to diabetes or obesity, is much more natural and different from what was done in 2 dimensions in just regular cell cultures. So some of the drugs that you could not test other ways or would not be meaningful to test in human samples are now can be tested in this. So in this system. So initially I would see that you could be able to test your favorite candidates in this type of almost a preclinical system for both efficacy, also as well as modes of action and functionality and safety, of course. For the more wide use, I would expect that you would need to miniaturize things in a higher throughput system where you could test multiple things. Now, I don't think this could be used as a really wide screen for thousands of compounds. But I mean, for the smaller targeted set of compounds, probably could be modified to be used.
But Pfizer, I mean, I don't know how much you can say, but Pfizer intending to bring something like this in-house, or they continue to work with Duke and University?
So far, they work with us at UK.
Well, thank you very much for that.